Good afternoon, everyone. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. And my name is Tailise Rodrigues. I am a postdoctoral research fellow at the University of Exeter, and I am also a member of the Society's Policy Advisory Panel. And I'm delighted to welcome everyone to today's webinar on antimicrobial resistance, taking effective action. The Biochemical Society believes that molecular biosciences play a key role in addressing societal challenges, including the ones related to public health. And antimicrobial resistance is a pressing example of that, isn't it? It threats to cause 10 million deaths by 2050 and to push 28 million people to extreme poverty. The session will follow on from our previous Biochemistry Focus webinar in 2023 and the Society's position statement on AMR, which highlighted the connection between AMR and molecular biosciences, the importance of the One Health approach for tackling the AMR threat, and the crucial need for innovation and new models of funding to maintain a pipeline of new antimicrobials. This webinar will revisit this crucial topic to further discuss and explore what it will take for action on AMR to be effective. And it's now a great privilege to introduce our speakers, Professor Anthony So, Professor Martha Clokie, and Misbah Khan. And before I hand over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar, but please do send your questions during the talks. If you have any question, please type in the question box as shown in the image on the screen now, and stating who your question is for, please, and we'll try to answer as many as possible at the end. Our first invited speaker today is Professor Anthony So, a distinguished professor of the practice and director of the Innovation and Design Enabling Access Initiative at the Johns Hopkins Bloomberg School of Public Health. The IDA initiative also serves as the secretariat for the Antibiotic Resistance Coalition, also known as ARC, an alliance with civil society groups and the South Center, an intergovernmental think tank of 55 developing countries. Professor So has served as co-convener of the United Nations Interagency Coordination Group on Antimicrobial Resistance, was part of the Antibiotic Resistance Working Group of the U.S. President's Council of Advisors in Science and Technology, and conducted research on reengineering how antibiotics are brought to market as a recipient of the Robert Wood Johnson Foundation Investigator Award in Health Policy Research. He was a co-lead author of the United Nations Environment Programme's Global Spotlight Report on the environmental dimensions of AMR in 2023. So I can see your screen, Professor So over to you.
Thanks so much, and thanks for the generous introduction, as well as the opportunity to offer a perspective on this panel. This discussion comes at a time less than a year after the UN General Assembly made its second high-level political declaration on antimicrobial resistance, and we are in the midst of a crisis in the financing of global health programs. While the US, of course, is cutting foreign aid by over half, significant reductions in overseas development assistance by other donor countries, including the United Kingdom, France, Netherlands, Belgium, and others, are also under consideration and could range from 25 to 40%. So the challenge we face in enabling effective global AMR governance and action is even greater today. Next slide. None of these recent changes, however, alters the reality we face in tackling antimicrobial resistance, or AMR. The Lancet Graham study reset upwards the estimates of mortality due to bacterial antimicrobial resistance, or AMR, to 1.27 million lives in 2019, a toll that exceeds the lives lost to HIV/AIDS, malaria, or breast cancer, as you can see on the graph on the right. Importantly, the study found that the AMR burden was greatest in sub-Saharan Africa and South Asia. Of note, in sub-Saharan Africa, where the AMR burden was greatest, the percentage of deaths attributable to AMR was lowest. However, the rate of deaths from infectious diseases in this region was so much higher that it lifted the toll in sub-Saharan Africa higher than any other region in the world. And this immediately tells us that while AMR is a global health challenge, the nature of that challenge and its solutions are different across regions. A context-specific strategy in sub-Saharan Africa, as opposed to high-income countries, would likely prioritize lowering the overall burden of bacterial infectious diseases, not necessarily just investing in developing new drugs for drug-resistant infections. Next slide. But like so many public health challenges, antibiotic resistance has had to wait for its day in the sun. Back in 2001, the World Health Organization laid out a global strategy for containment of antimicrobial resistance. But they could not have picked a more inauspicious place and date to announce their report. As David Heyman, then WHO Assistant Director General, tells it, they were set to do so on September 11, 2001 in the Washington Press Club. Needless to say, the press conference was canceled as one of the hijacked planes crashed into the Pentagon, and activity on antibiotic resistance was near moribund thereafter at WHO for several years. But today, the issue of antimicrobial resistance is the focus of a global action plan adopted by the World Health Assembly in 2015, followed by the Food and Agriculture Organization and the World Organization for Animal Health. It has garnered the attention of the G7 and the G77, and it has come before a high-level meeting at the UN General Assembly twice, once in 2016 and then again in September of 2024. Along the way, many have played a part in awakening the world to this global public health threat. Next slide. In 2016, our program partnered with the UN Secretary General's office in New York to host a policy briefing on meeting the intersectoral challenge of antimicrobial resistance in June of that year. And this was in lead up to the UN General Assembly high-level political declaration on AMR that September, only the fourth time ever that a global health issue had come before the UN General Assembly. That declaration called for the creation of the UN Interagency Coordination Group, or IACG, on AMR. As one of its co-conveners, we delivered our recommendations to the UN Secretary General in 2019. Now, importantly, the IACG recommendations called for the creation of a tripartite joint secretariat, comprised of the World Health Organization, the Food and Agriculture Organization, and the World Organization for Animal Health. Next slide. Now, the IACG recommendation laid the groundwork for establishing the tripartite joint secretariat, but by 2022, at the urging of a number of us, the UN Environment Program was added to make the tripartite a quadripartite secretariat. UNEP's addition made the global efforts truly one health in approach. The IACG recommended the creation of 3 other entities to round out global AMR governance. The Global Leaders Group was established, but has not been so successful in mobilizing the resources for global AMR work yet, but has importantly, of course, raised awareness of the concerns over this threat. And later, the multi-stakeholder partnership platform diagram on the right-hand side of this slide came into being, but its consensus-based approach and very diverse membership from industry and academia to governments and funders has made it challenging to become a platform for catalyzing much new change. The third entity proposed by the IACG was the Independent Panel on Evidence for Action Against Antimicrobial Resistance. It has not become operational, but its creation was again called for by last year's UN General Assembly's high-level political declaration. However, 6 years after we first proposed it, it may no longer be as relevant. With 4 international agencies now coordinating their work on AMR, the value of independent expert guidance is a little less clear. How would it substitute or complement for what the quadripartite agencies are already doing? Will it distract or take resources away from their already underfunded efforts? And most importantly, what topics would it tackle and catalyze to make a value-added difference? So how do we make AMR global governance actually work beyond the now periodic exhortatory declaration? Let me suggest that we might do better. To commit to measures and targets. 2, to engage and enlist civil society and private sector partners. 3, to integrate AMR into broader development health concerns. And 4, making the case for investing in AMR. Next slide. Unlike other disease areas from HIV/AIDS to TB and malaria, we have been slow in arriving at global goals by which we might track accountability on AMR. Up until a few years ago, the Sustainable Development Goals had not a single AMR-specific indicator to track progress on this global health challenge. We now have one on stewardship of bloodstream infections in hospitalized patients. In 2022, the 3rd high-level ministerial conference on AMR resulted in 47 countries endorsing the Muscat Ministerial Manifesto on AMR. These countries committed to quantitative targets for both reducing antimicrobial use in the agri-food system and to a health sector target as well. Reducing agri-food system use of antimicrobials by at least 30 to 50% by 2030, zero use of medically important antimicrobials for human medicine in animals for non-veterinary medical purposes or in crop production. And in the healthcare delivery system, WHO had classified antimicrobials into three categories: access, watch, and reserve. Access antibiotics are meant to be the most broadly available in the healthcare delivery system. So the third quantitative target was in the healthcare delivery system and called for ensuring that access group antibiotics comprise at least 60% of overall antibiotic consumption in the healthcare delivery system by 2030. While the UN General Assembly in 2024 embraced the access group antibiotics target and upped it to 70%, the food system targets signed by those 47 countries in the Muscat Ministerial Manifesto were not adopted, nor were they supported in the fourth global high-level ministerial conference in Jeddah, Saudi Arabia last November. The UN General Assembly's high-level political declaration, though, did lay out some other targets, among them a 10% reduction in global AMR-related deaths by 2030, but only the aspiration of $100 million to help 60% of countries to fund their national action plans by 2030. However, we have a long way to go, with only 11% of countries reporting that they have allocated funding and are actively implementing their national action plans on AMR. Next slide. Now, how do we enlist and engage civil society and private sector actors in tackling the challenge of AMR? Civil society groups like the members of our Antibiotic Resistance Coalition have worked to influence food procurement so that those buying for restaurant chains or grocery stores would do so responsibly and insist that the food animal products they source be raised without the routine use of antibiotics. Beginning in 2015, a group of US-based NGOs issued the Chain Reaction Report, calling initially on the top 25 restaurant food chains in the country to do just that for poultry. And over the first 3 years, this consumer report card prompted first 5, then 9, and then 14 of these top restaurant chains to make changes and steps to limit antibiotics in their supply chain. And they accounted for 2/3 of all fast food industry revenue in the United States. This had ripple effects upstream on the Tyson and Purdue's in the poultry industry, but pork and beef had lagged behind. The Center for Science and Environment in India would put out its double standards report. This report noted that the timetable for multinational companies removing antibiotics from their supply chains in the United States did not extend the same to India. In the United Kingdom, the Alliance to Save Our Antibiotics put out a consumer report card on UK grocery store chains and how they were managing these practices in their own branded products and those stocked up from other producers. And there are many other examples, from testing of retail food products for antibiotic residues to drug-resistant microbes, that have helped to raise consumer awareness in these public-facing brands and sometimes greater accountability through their food procurement practices. Food suppliers, too, are responding to growing consumer demand for healthier, safer foods, from organic certification to even a number of Ecuadorian shrimp fisheries that have banded together under the Sustainable Shrimp Partnership. The Sustainable Shrimp Partnership has worked to blockchain certify shrimp from these Ecuadorian shrimp fisheries that have committed to zero use of antibiotics, adhering to standards that exceed even those of the Aquaculture Stewardship Council. So we are seeing market signals, both on the supply and demand side, working to reduce the use of antimicrobials in the food system. Next slide. How might we better integrate AMR into broader development and health concerns? Well, there are many opportunities, from ensuring effective vaccine delivery to water sanitation and hygiene or WASH interventions. The example I share here is the case made by the Organization for Economic Cooperation and Development, or OECD, comprised largely of high-income countries. The OECD put out a report in 2018 that showed that AMR containment measures could be cost-effective. In OECD countries, a comprehensive intervention package that addresses AMR both in hospitals and clinics through improved hygiene and stewardship, and also in communities through delayed prescriptions, mass media campaigns, and rapid diagnostic tests, could come to just $2 per capita a year. Such an investment, they projected, would avert 47,000 deaths a year in OECD countries, and the public health package could pay for itself in under a year and save $4.8 billion a year in OECD countries. In low and middle income countries, as opposed to high income OECD countries, the AMR package might take a different form, perhaps a greater linkage to WASH interventions, or improved vaccinations, or the targeting of AMR hotspots in the urban environment. Putting together a compelling, cost-effective, and even cost-saving package would make a huge contribution to country-level efforts to address AMR. Next slide. Disappointingly, though, the UN General Assembly's high-level political declaration 2024 did little to step up global commitments to finance efforts to tackle AMR. It set an aspirational goal of $100 million for the quadripartite efforts to support national action plans by 2030. Much of this work could be supported by the UN Multi-Partner Trust Fund on AMR, but that fund remains, over 6 years after it was established, under-resourced, with just over $30 million in commitments, barely a rounding error to the billions called for by COVID and by climate change. Even before the pandemic, the World Bank projected that if AMR went unchecked, up to 24 million more people could be forced into extreme poverty by 2030. It looks like COVID-19 might have beaten AMR to the punch there, but under the high impact AMR scenario, it could amount to a 3.8% loss of annual GDP by 2050, with an annual shortfall of $3.4 trillion by 2030. The bank had concluded that, quote, putting resources into AMR containment now is one of the highest yield investments countries can make, unquote. By investing just $9 billion a year in low and middle income countries, we could avert trillions of dollars of potential economic losses. Significant returns on this investment would accrue to low and middle income countries, but take a look at the pie chart on the right side of this slide. Over 80% of these returns would benefit upper middle and high income countries, the very countries that would be best positioned to make this global investment. Next slide. So I hope this brief overview provides several concrete approaches by which we might make the case for more effective global AMR governance. COVID-19 may have been like a tsunami, while drug-resistant infections are like a rising tide. However, the need to act on AMR is as urgent. And even though the UN General Assembly high-level political declaration suggested a global consensus, it is the day after and every day after when the real work must begin to transform principles into priority actions. And updating the Global Action Plan, one of the key goals on the agenda this year, and taking some of the approaches I've outlined in this talk will be critical to that. We delve further into the approaches we touched upon here in our commission paper for the South Center, Catalyzing Policy Action to Address AMR, Next Steps for Global Governance. The tiny URL on the slide will take you to that paper. As in climate change, the choice is clear. We can pay now to address antimicrobial resistance or pay much more later. Thank you for the opportunity of sharing these thoughts with you.
Thank you, Professor So, for such a forward-thinking talk. And now our second invited speaker is Professor Martha Clokie, Professor of Microbiology at the University of Leicester and the Director of the Becky Mayer Center for Phage Research. Professor Clokie is also the Editor-in-Chief for the journal Phage, Therapy Applications and Research. Her work involves the identification and development of bacteriophages that kill bacteria that causes diseases in humans and animals in an effort to develop new antimicrobials. Her work and that of the Beck-Mayer Center encompasses all aspects of phage therapy development, from unraveling fundamental biology to product development and commercialization. Martha's work is regularly and recently featured on the BBC, for example, on the In Our Time, Bacteria, the Tiny Giants, the Life Scientific, and the Infinite Monkey Cage, and the Today program as well. So I can see your screen, Professor Clokie, and over to you.
Great. Well, thank you very much for the invitation to be here. It's really exciting to be part of this today's seminar on this incredibly important topic. And my seminar, I hope, follows on nicely from our last speaker, in that I'm now going to delve into the specifics of one very promising approach for bacteriophage, for antimicrobial resistance, which is to develop bacteriophages. Now you can see on my very first slide that antibiotics were actually discovered after bacteriophages. So we know that bacteriophages were formally identified in 1915, but they were actually much more complicated to develop as antimicrobials, given that you have a large, complex, replicating organism instead of just having a simple compound. So what I've drawn here just to sort of represent this is that as soon as antibiotics were discovered, actually, they were much more quickly developed as antimicrobials. And however, due to their overuse, as we heard, both within the animal and the human food sectors, as well as in medicines, they've really been abused to the point where they are no longer working. So we're just seeing increasing number of bacteria that we just can't treat anymore. So now, although bacteriophages are more complicated to use, it's this complexity that we now need. For example, bacteriophages are exquisitely specific. So each bacteria that causes us problems, for example, E. coli, Salmonella, Campylobacter, they all have very, very different, exquisitely different bacteriophages. And the phages are not just species specific, they're often specific to the actual strain within a species. So this necessitates the need to have large numbers of bacteriophages. So what I want to do today is talk a little bit about this research, tell you about how we would use it, and the three pillars of the work that we're doing within our Becky Mayer Centre for Phage Research. So I thought, therefore, I would, next slide, please. I thought, first of all, I would tell you about the centre itself. So we are an actual centre rather than a virtual one. We opened a couple of years ago. And then we've actually had some really quite major investment, which has allowed us to now map our vision to what we can actually do. So we have, last year we had some a £4 million investment from a philanthropic source. We also had major funding from the BBSRC, which is the British Biology Research Council, and also charitable funding, as well as we're leading a grant from it's called the Joint Programme Initiative for Antimicrobial Resistance. So this is a very nice €1.8 million grant that brings together us with major labs across Europe, so in Poland, France, Germany. It's also an Australian lab involved as well, and a Swedish lab, as well as partners in a hospital in Bristol. And the idea of this grant is to try to do all of the specific things that are needed with bacteriophages to actually be able to translate a product. So we've got the very best teams looking at formulation, about resistance, about delivery, as well as immunology, and then the pharmacodynamics. So it's not totally trivial to translate bacteriophages into products. It takes a lot of different types of expertise. So this is really what we're trying to do within our phage centre, is join things together. We're also trying to establish a very well curated bank of phages to be able to actually have that biological resource to choose from in the first place. And then we're working increasingly with our regulators to make sure that the correct data is taken for to be able to regulate a product and therefore get it to the market. So just one slide here to say that if you want to hear more about the history of phages, please feel free to listen to this in our time. It's a really lovely episode. It was great fun to be a part of recently. And they worked with a phage historian there who really talked about the very early turbulent times of phage discovery. The main thing I want to show you from this figure is that both Fort, the Englishman, and Durrell, who co-independently discovered phages a couple of years later, they then trained a young Georgian scientist, Georgi Eliava. So if you have an idea of phages being something that's a bit associated with Georgia, Russia, this was the connection. And what Eliava instigated is this institute here. He established that as a major source of phage research, which has continued to this very day. So phages have got a long history of being used. However, what we can do now, and it's important within this biochemistry context, is we can go straight from identifying bacteriophages to really understanding the genomic contents. So although they've been worked on and people have been trying to develop them or developing them for 100 years, we can now do this in a mechanistically informed way. So it's really should unleash a whole lot of new effective products to give us another chance at tackling bacteria. So what you can see on my slide here is, if you might recognize your favorite pathogen of bacteria if you have one, in each dot is a bacteriophage genome. What I really want you to see is, if you look at these individual clouds or clusters of phages, if you look at Salmonella, in Klebsiella, where my cursor is, you can see that there's nothing in common between this group and this group and this group, but where there is some bit of connection, that means that if you look at the right side of the Klebsiella, there is connection between those phages, which shows that there is some genetic relatedness. So each group has different sets of bacteriophages that target it, and there can be up to 20 or even more groups of phages that literally do not have a single recognizable gene in common. Okay, so phages are the most abundant biological entity on the planet, and they're also the most genetically diverse, which brings with it whole new biochemistries and chemistries and biologies and different ways of doing things in terms of understanding how they actually work, how they kill bacteria. So it's a very fruitful area to invest in for that reason. So we spent a lot of time here with their discovery. So I want to just make the point here. Can you see in this figure, there's these large particles and all these tiny little particles. They essentially are the bacteriophages. So for each bacteria anywhere, there's thought to be 10 phages. And what's different now is the speed by which we can get that genomic information is massive. So we can go straight from finding a bacteriophage, and you see these little dots on these are lawns of these are individual phages that have gone in and killed bacteria, to the point that we can now see their zone of killing on a plate. We can take an individual phage, literally one of those tiny little plaques, and we can sequence it now for about £5. Just takes a few days as well, literally 3 or 4 days for the whole process. So this massively changes how we work with phages, because it used to be we'd isolate them, we'd do lots and lots of biology experiments to work out which were interesting, we'd bulk them up, and then we would look at that genomic data. But no, we can get that now using GridION or nanopore technology to be able to get those genomes. So you might think, why do you need so many bacteriophages? Why are they isolating all those phages? Surely there must be enough isolated, but no. Say, for example, if you want to work on killing E. coli. So E. coli can cause infections in all these areas of the body, and depending where they are, the phages will need different properties. The bacteria will be different, the pressures on the phages will be different, so temperature, pH, biofilms, and so on will be really, really important. And then how they work in oxygen, salts, and airflow, and also in ultraviolet light. So therefore, in order to be able to find phages with the right properties, we have to identify a whole lot in the first place. And also, I think it's important to make the point that phages can be used together with existing antibiotics, so they'll need to work. And we know that in some cases there's very, very good synergies. So this is another thing we look for. In terms of research, when we got this big body of phages, we do many things to try and figure out why they work, and largely this involves working in much more, in the actual environments that we'd like to work in. So I thought I would tell you about one story just quickly, which I find very interesting within our Salmonella phage work, because we were able to do some cryo-electron microscopy that's linked to some phenotypic behaviors that we saw with our bacteriophages. So this is showing you a phylogenetic tree of a highly, highly related set of phages that all kill Salmonella. Now, what was interesting was some of these phages are really stable. So normally, if you heat a phage to more than about 50 degrees, it falls apart. But actually, just some of these phages with very small differences were stable up until 90. So this would mean you'd be much better at being able to formulate your phage to a stable product. And what we noticed when we looked at the genomes of these phages to see where the differences were, they both coded for glycosyl hydrolases. And these are proteins that are thought to be involved in adding sugar groups to proteins. And then recently, we got around to looking at the bacteriophages according to cryo-EM. And can you see this cloud around the head of the phage? That is typical of a glycosylated picture of a protein. So what we think is happening is the phages that are stable are indeed having their heads glycosylated. So I'm just showing you some more detailed images now of the different components of this phage. So what's I'm really trying to make the point is that having access to this advanced imaging as well as to genomic sequencing can really help us accelerate the pace of translation to get these mechanistically understood bacteriophages. We're also increasingly working with our end users, for example, doctors, to do things like, can you see this bottom right picture? This is looking down a catheter. So this is a catheter that was being used to catheterize somebody with a bladder infection. And unfortunately, these catheters themselves can get really coated in bacteria. So we've been working to see, well, could we use phages actually to coat the catheter in the first place to prevent infection in this context? So we're increasingly working with our end users, and this is some data from this. We can see these are the untreated catheters, and then these are the treated catheters with phages. We can show that phages work really, really well at chewing nasty biofilms from patient samples. We've also been looking to see how we might develop bacteriophages to use as products in humans. So we have a mouse study, a mouse model where we can infect the mice in a stable way, we can collect urine, and we can show that just one dose of phages, can you see by the end of the week experiment, has completely cleared away the infection. We've also shown, you can see by looking at these green bars, that the bacteriophages do actually replicate in that bladder of the mouse. So we can see that it's very much of a romp through, I'm afraid, but hopefully I'm really trying to show you the fact that investing in bacteriophages means we've got this product that works in a different way, they replicate at the site of infection, they're highly effective, and we can use this genomic data to actually see how they're working. So hopefully I've really tried to show you that it's really a very exciting time to be investigating and developing this technology. We can bring this long history of use that I mentioned into a sort of mainstream, mechanistically informed practice. We do need to have these large collections of well understood phages, and also we need collaborations with all sorts of different types of scientists, particularly biochemists. Our biochemistry collaborations that are the most effective. I'm actually currently giving the seminar from NIH, from the National Institute of Health, where I'm working with a very, very good phage biochemist here. And also we need these key partnerships with our end users to be able to pull bacteriophages through. So with that, I will end my seminar. I'd just like to thank the lovely, hard-working people that produced the data that I showed you, who are featured in this little picture here, and to thank the funders. Thank you for your attention, and happily look forward to discussing with some of you some questions at the end of the seminars today. Thank you.
Thank you, Professor Clokie. That was very insightful and brings us hope for new treatments, right? And now is our third invited speaker is Misbah Khan, a health and environment reporter specializing in antimicrobial resistance at the Bureau of Investigative Journalism. And Misbah has previously worked on investigations for The Guardian, ITV, Channel 4, Channel 5, and more recently she has been working on the Wounds That Won't Heal project with the Bureau of Investigative Journalism, which aims to document the stories of people from around the world that are directly affected by the growing threat of AMR. And yeah, if you would like to share your screen, and then we can, I can see it, so over to you.
Amazing, thank you so much. Thank you, speakers, for having me to discuss the communications and engagement side of antimicrobial resistance. As many of you can imagine or have been through your own experiences, AMR can be quite a complicated topic to translate to a lay audience. But I'll give a brief introduction to the organisation I work for. So I work for the Bureau of Investigative Journalism. We're the UK's biggest non-profit newsroom, and we work independently to cover issues such as antimicrobial resistance. I'm the Bureau's health and environment reporter. I've got a biomedical science degree and have accidentally ended up in journalism. But so as soon as the AMR reporter role at the Bureau opened up, my previous sort of academic experience came to use. So as Tailise mentioned, I've been covering antimicrobial resistance for the last 3 years, and around the UN General Assembly meeting that Professor So mentioned earlier in his presentation, we were actually commissioned a few months earlier to do a photography project that sought to bring antimicrobial resistance alive, so and the human stories to the center of the conversations that were happening at the United Nations. And so we traveled to a number of countries. I could try to dot them quickly on this map here. So we went to Pakistan, Ethiopia, Nigeria, and a couple of places in the US to see how antimicrobial resistance really manifests itself on the ground level. And although I'd been covering it for a while, I was still surprised as to what I witnessed on the ground as well. I should give a content warning. So some of the photographs that I'm about to share are a bit distressing. So I'll start you on my journey in northern Pakistan, where I met with an 8-year-old boy called Akasha and his father, who's in that photograph. Akasha's father had told me that both Akasha and the child living across the road had been bedridden for months with drug-resistant typhoid. The boys had been left weakened by headaches and fever, and the father suspected the water system in their school had been contaminated, which meant children across the entire village were ill. And then sort of the next little girl that I met is Misbah Khan. She was visiting a free clinic in front of an Afghan refugee settlement, again in Peshawar, where she lived. The doctor suspected that the 2-year-old had drug-resistant diphtheria. She was on critically important antibiotics. In other contexts during my reporting, I saw that these drugs were handed out like sweets. We followed her to a home where I discovered a number of the family members had the same symptoms, including her 3-year-old cousin, but they couldn't afford treatment for Furqan, and only Misbah Khan ended up getting the drugs that she was needed. In the same clinic where we met Misbah Khan, there was also a 6-year-old Ariba, and she had sandworm disease. A wound then opened up on the back of her head, and her head had gotten infected. The doctors again had tried a number of antibiotics that weren't working to treat her wound. We also visited local hospitals, tertiary care hospitals, where Malik, I met an embroiderer named Malik who had traveled from another province where he had failed to treat his infection. The next image is a little bit, well, not a little bit, it's actually quite distressing. So just a content warning before for viewers. Malik has cut his foot whilst crossing the road, and his wound had become infected, and his unmanaged diabetes meant the wound developed into gangrene. The drug-resistant infection had spread, so by the time he reached the hospital, he needed his toes amputated. In southern Pakistan, a place called Thatta, we met Amir, who used to work in the dairy industry in Karachi. Whilst living in the slum in Karachi, he contracted TB, tuberculosis. His illness meant that he was no longer able to work and had to return home to a patch of rural land. A few months later, Amir's brother Rab got drug-resistant TB. Although they were all receiving treatment from the district hospital, the brothers were only well enough to be able to pick up small jobs around the village and had to resort to begging. Despite conditions already being hard, they were also looking after orphans their older brother had left behind after having died from complications of diabetes, which really demonstrated the socioeconomic impact that something like drug-resistant TB can have. After that, I went to Lagos. Sorry, there's an extra slide. So I wanted to demonstrate also how these stories then ended up in the news. So we partner with other news publications, and during the UN General Assembly in particular, and the date of the assembly, we actually published this story, and you can see our media partners on the side there. So we published with local Pakistani news outlets, including Dawn, The Guardian also, and The Lancet, for a more specialized audience that had insight into the types of drugs we were seeing and the testing that was done. So we got pretty good coverage of Akasha and Abu Zaher's story into mainstream media outlets to build up that momentum that we're looking for, and hoping that policymakers that aren't necessarily buying into AMR or funding AMR would then start taking the issue more seriously, beyond seeing it as a number of acronyms and another global health issue. So this was the Lagos story that I was going to go towards. So in Lagos, Nigeria, you can see here how dark it is because there's no electricity. This is an ward for newborn babies at the Children's Hospital, but during the frequent power outages, the incubators that the babies are in don't work. Most of the babies, like Ahamba, were born outside of a hospital to a traditional birthing attendant. The ones who were brought here almost always had sepsis. Ahamba was on a last resort antibiotic, meropenem, which is only used when other drugs don't work. The hospital bills in Nigeria are covered by the families, and so babies like Enioha, who had also had drug-resistant sepsis and was on third-line antibiotic, she also had other complications, and very sadly, her parents had abandoned her at the hospital after they were asked to pay the bill. Babies like Enioha, though, rely on nurses like Ibazim to survive. They work to ensure babies are kept warm during the blackouts. And then the next stage of the story, I mean, I think this was probably the hardest reporting trip I've done. The Amhara region of Ethiopia was in a state of emergency when we visited because of the ongoing conflict there at the time. Hospitals had been raided by warring factions, stealing vital medicines, which means babies like Rahmat had a slim chance of survival. And so Yusra is another baby that we met who also had drug-resistant sepsis. Yusra's twin had already died at 6 days old. The hospital didn't have labs needed to test what infections the patients have, which means life or death decisions on which antibiotics to use was essentially guesswork. The first two they tried didn't work, but with babies this small, they were against the clock of survival. Both newborns needed last resort antibiotics, which is expensive in comparison to an average salary. Neither family was able to afford it, and they took it, they had to get it, they had it in stock in the pharmacies nearby, the hospital gate, which we saw, but Yusra and Rahmat's families couldn't afford it, and by the time we'd returned to the desk, both had actually died. Then we'd sort of use those stories again to publish it with local media outlets, including the Reporter magazine in Ethiopia, Premium Times in Nigeria, the Globe and Mail and actually in Canada, and the Guardian here and internationally, to get again coverage of AMR. If you can see, like all of the AMR coverage we've done so far is always tied into a bigger issue or another issue, because AMR on its own, in terms of statistics and sort of other things, doesn't necessarily get the buy-in or interest of readers or even my editors, to be honest, when I'm trying to pitch a story. So another issue we tied antimicrobial resistance to was UTIs. And so when we went to the United States, we met with Tamara, who had been suffering for 10 years with a urinary tract infection, which had been increasingly unresponsive to antibiotics. And without an accurate test, she had tried every treatment possible to try and relieve the excruciating pain, a pain so bad that she had tears in her eyes when recalling it. Her attempted treatment, I mean, included various antibiotics, herbal supplements, surgical procedures, and medicine that she had administered to herself through a catheter. She still lives in fear of her near infection, the last time I at least spoke to her. And sort of, you know, the stories that I have shown you are, so we published that story again, but this one didn't necessarily generate partners. Again, that was more for logistical reasons than anything else. But we'd got it, like every single time I was telling someone at the time of reporting what I was covering, they always knew somebody that had sort of UTIs that they were struggling to get rid of, and didn't necessarily understand why. And people, if I mentioned AMR to them, they wouldn't know what it was, and they would just think the doctor doesn't know necessarily how to treat their infection, so they would go to another doctor rather than realizing what they might have been dealing with potentially. So this story really resonated with a lot of people that had read it. They either knew someone themselves or had gone through it themselves, especially if they had female anatomy themselves, because women are 30 times more likely to get a UTI than men are. But yeah, you know, there were a number of stories that we collected whilst we were out there. I've only named a few, and one of the shocking things about this was how easy it was to find, in fact, people with experiences of these crucial drugs not working. And you know, we also saw themes of sort of prescribing practices, the impact of conflict and climate change, and hoping to continue working on this project and to keep up the communications and engagement side side of antimicrobial resistance and allow policymakers to engage with the topic a bit more. The photography has been shown as an exhibit on its own in and around high-level meetings like this UN Assembly, but also others where we're trying to get people to invest more, including Saudi humanitarian conference that happened recently this year, and we're planning to also pitch around the Nigeria ministerial that's about to happen next year in July. But yeah, I would like to thank all of the individuals who have consented to sharing their stories for the project so far. We had a robust consent process to ensure they were happy with their photographs being taken and their stories being told. And so it was a lot for them to share the most vulnerable sides of their stories. Thank you.
Thank you, Misbah Khan. This is really thought-provoking, isn't it? And it's really sad. Makes us really want you to be able to do something quickly. Yeah, so now we can welcome questions for any of the speakers, and please keep sending your questions through the question box as shown on the, it's going to be showing in the image on the screen soon. So yeah, I have a few questions already here. And the first one is for Professor So. It seems the investment case for tackling AMR is clear in terms of costs. That will save in the long term. Is this long-term view enough as an incentive for governments to act?
We wish it were, but as in climate change and so many of our public health issues, it is not. And I think it's a really important issue because oftentimes I think we are all susceptible to, particularly among policymakers, to hyperbolic discounting, which is where policymakers disproportionately prefer immediate rewards, even if waiting actually yields a better outcome. So we frequently discount actually the investments necessary to make those changes today that have much greater reward down the road. And we, of course, we can see this in a number of public health areas. We also need to take these global economic estimates down to the country level because the return on investments, we're competing against many other areas to actually, of course, beckon for attention, and many of them very deservingly so. And so when we try to make the case for a vertical issue like antimicrobial resistance, it's challenging. So we need to figure out how to really integrate our concerns to be shared ones with other areas: maternal and child health, vaccination, wash interventions, water, sanitation, hygiene interventions. These are also very important, of course, global health challenges, all of which deserve attention. And if we can somehow make the case that some of those efforts will also translate into gains, actually prevent and lower the burden of infectious diseases, and thereby also the use of antibiotics, and thereby reduce, of course, the turn the tide perhaps on antimicrobial resistance emergence, that would be really important. We also need to re-engineer the healthcare delivery system, because in many ways we do not have a system that incentivizes sufficiently those who are positioned to make the changes like antimicrobial stewardship, or those in the food system who could see the market signals and respond appropriately. But we need to actually build that connections more strongly. That will take both civil society, private sector, and governmental action to work together. So for us to really realize the sort of financing that we need to see in AMR, we're going to need to do all of that and make the economic case too, so policymakers will actually act, because they do move. I think they're moved a lot by the stories, of course, that Misbah Khan actually so brilliantly actually described and powerfully portrayed. But we also need to then translate into how we actually can use the limited, essentially, economic and financial resources we have, so that we can make the opportunity cost case for AMR as being paramount.
And if I could follow up on the same question and ask one myself, so do you think more awareness from the population about the AMR threat, especially in, I would say, middle-income countries, because, the low-income countries, they are really feeling the effects. In high-income countries, we are having the chance to discuss this, but in general, more awareness would help increase the pressure.
Is that to Misbah or to me?
To you, sorry. Just following up on what you said now, and then, it also could be for Misbah as well. It relates to her work, but I want to hear your thoughts.
I think in order to convert awareness into action, the level of awareness is growing thanks to the important work of many who are telling the stories. And of course, there's a whole new task force on for AMR survivors that WHO has in fact created. And that has been very helpful because for years, we've oftentimes said that, AMR seems like it's a problem with statistical victims. It doesn't have that kind of actually ability to mobilize like, Mothers Against Malaria or, HIV/AIDS, where there are clear, target groups that actually can mobilize and actually speak to it within the healthcare delivery system. It just seems somewhat more diffuse. And with over half of the antibiotics by volume being used, of course, in the food system, it is a much more challenging One Health issue. And we need to find a way, of course, not only to tell the story, which does increase awareness, but we have to really connect those stories more clearly to concrete strategies, because it is not enough to simply, of course, be concerned that in fact there are deaths, but what do we do about it? So for example, if you look at the estimates that came out of the Lancet Graham study that were then projected in September of last year to look at the 2025 to 2050 time window, it turns out that the best investments in terms of return would be in fact towards actually better care. They have a package comparing it to gram-negative actually drugs. If we have the gram-negative drugs, we might avert 11.1 million deaths in that time interval. But if we actually had, in fact, better care packages that they actually were describing in this modeling study, we would avert over about 92 million, if I remember correctly, deaths in that interval of associated and attributed to AMR actually deaths. That's 5 times difference there. So how we use those stories to direct us to the right actually policy interventions. I'm not saying those are mutually exclusive, of course, policy options. I think we need to do both, obviously for the treatments as well as obviously the care. But we do need to actually particularly understand the context in middle income countries and in lower income countries, what the priority needs to be today in order to actually get policymakers to act.
Okay, yeah, thank you very much. And Misbah, I don't know, since it relates to your work as well, do you want to share your thoughts on this question too?
Yeah, I mean, it's been really interesting doing this photography project and translating it to people and ministers. So you have like individual policymakers that then come up to you and realize that, oh, this is how this is impacting me in my country, and who necessarily wouldn't have engaged it outside of the the diplomacy circles that exist at either the United Nations or the WHA. So it's like sort of showing them that reality. So one particular example was when I was in Pakistan. I had I had the time and ability to speak to a lot of the ministers that were involved at like sort of more provincial and local level, as well as more on the high up, federal level. And so it was something that wasn't even on their radar, even though it's a massive issue in Pakistan, because they're not engaging with it. So you have like the international meetings, and then you have the local context, and those don't necessarily connect in the way that you would hope that they would. So I do think more context-specific and localized awareness is important amongst people like district health officers and whatnot, and they can drive their own solutions to the problem in themselves as well. Once they realize, like, in different contexts, in Malawi, I've heard that disposable oxygen tubing is a massive issue in terms of Klebsiella outbreaks on neonatal wards. The fact that it's disposable because they run out of shortages, and so you have the choice between having a baby that you need to give oxygen to, but reusing the plastic tube that's been used in another baby that potentially has sepsis, but you can't confirm because the lab results are taking too long to come back. And in the moment you need to give that, you reuse that oxygen tube without necessarily doing the best you can in terms of sanitizing it. So like that's the context that people are working in, but you don't necessarily have WHO guidelines on how to sanitize single-use items because you don't want to encourage that practice. But that is a very lived reality for a ward in Malawi, and it's a very context-specific. I guess the point I'm trying to get across is context-specific awareness amongst local policymakers themselves needs to be a very real thing that I think we need to do to create some active change, because you have two very separate conversations happening, and I don't think they don't meet very well, even though everyone's very well trying to tackle the same issue in terms of saving lives.
Yeah. Oh, thank you to you both for your thoughts on that. And I have a question for Professor Clokie now. When you sequence and characterize a phage, what are you looking for to determine if it's a useful therapy?
Well, ultimately, what we want to be able to do is predict which groups are going to be the ones where we can put more investment into doing the research. And largely what we're looking for are bacteriophages that kill quite broadly within a group. So Klebsiella that was just mentioned by Misbah. There's a lot of different types of Klebsiella that have different capsules. They have different, the outside of those bacteria are quite different. So you need phages that can kill a whole range. So you look for a breadth of killing, and we're also looking for those phages that can kill more aggressively. So not all phages want to completely take out all of the members of that that they can kill. They kill in different ways. So we're looking for the more aggressive, more virulent ones. And one of the things we're getting better at doing is predicting what patterns in the genome would have those features. We're also looking for things that might make a phage potentially that might make a phage not helpful in this situation. Like we know that some phages are capable of some behaviors, like perhaps even moving antibiotic resistance genes about. This is a very unusual, not many phages can do this, but we know the features of phages that can. So we're looking for both positive and negative attributes of bacteriophages that we would then focus on.
Mm-hmm. Oh, thank you. And I have another question for you. So what engagements and interests have you had from policymakers, decision makers on phage research and treatment?
Yeah, that's again a very good question. Until recently, I would say very little. I think phages just seem as being something that's a bit niche and not really relevant. But I think as people realise the severity of this, then that there is engagement now. So within the British government, there was an inquiry that was run 2 years ago in the House of Commons, looking at whether or not the British government should invest more in bacteriophages. The answer was yes. They produced a really nice report, and this was followed up by the House of Lords. They also gave evidence at that hearing. That was this January, and there have been tangible differences. I'm pleased to say that now phages are mentioned, at least in the UK strategy document for AMR. So we're gradually making traction. I think in general it's still not. I think in a way, I think it's quite hard to sell bacteriophages. It's quite complicated, isn't it? First of all, you've got to tell people about the difference between a virus and a bacteria, and then you've got to talk about define AMR, and they don't really understand that. So I think the messaging and the language that we use around phages could definitely be improved. But I think that the real need is definitely motivating a much bigger clinical interest than there's ever been, and that is feeding through now to policy makers in different places. So I went to a meeting in Uganda actually this year which was full of there was really good interest, for example, from people quite high up within the Ugandan government, as well as people at a more local level. So gradually we're making more traction, but we definitely could do with more helping communicate, making people aware.
Okay, thank you. But good to hear interest is increasing from policymakers and decision makers. Yeah. And I have a question for Misbah now. Do you think the upsetting nature of AMR and its impacts help or hinders getting the message about its importance across to the general public?
I've only seen it really help. It's never really shut down a conversation. It's always led to more inquiry, if anything. So I also do reporting on food and agriculture and antimicrobial resistance and overuse of antibiotics. That can be often difficult because it can lead to a lot of scaremongering and misunderstanding as to how antimicrobial resistance works in the food and livestock setting. But and it can lead to a sort of uproar. We did an investigation into Polish chicken products coming into the UK that had previously been associated with the drug-resistant salmonella outbreak, which did lead to people dying. And then the sort of inquiries we got were, do we need to throw our chicken nuggets from our freezer out into the bin? So it's kind of, but at the same time, it's a question that we tackle all the time inside the newsroom. Do we need to overcorrect the lack of urgency in the matter? Is it useful for people to know? But so it rises and then dips in terms of understanding. You very different populations have very different understanding of AMR. So it really does translate differently amongst different people. I've never seen it hinder the conversation for the most part, at least, or no one's at least vocalized it to me. So that might be biased on both sides in terms of people coming back to me with the feedback.
Mm-hmm. Okay, yeah, it's good to know, because it's important. It's scary, but it's important to keep the conversation going. Yeah, that's good to know. I have a question now for Professor So. Is there more organizations like World Health Organization? Actually, sorry, is there more that organizations like the World Health Organization could be doing on AMR governance?
So much more. It's not because they're not trying, it's that the financing actually for this area is remarkably not proportionate to the level of the burden of disease we are witnessing. And that's really, of course, what's so challenging, I think, for everyone in the space on this policy landscape. Now, I think that, of course, the quadripartite agencies are key in having, and part of that's the structure of global governance that's been really helpful to put together, but it's taken us quite a number of years since that 2001 initial WHO report, as I mentioned earlier, right? I mean, we are now a quarter century afterwards, pretty much, right? Almost, and we have to ask ourselves, how long will AMR wait, frankly, for us to actually develop a sustainable, robust response across sectors. And this is what makes it a really wicked problem. It is an intersectoral challenge. It is one in which the business model of the pharmaceutical industry, the traditional pharmaceutical industry model, doesn't work. If you sell more drugs, you drive more resistance, right? If there's greater consumption of drugs, then you inevitably drive more resistance. That's just that the biology and the economics essentially are at odds. And so we need to figure out ways in which actually to overcome all that. And that makes, of course, it very challenging for each of these, of course, international organizations to take such action. I think, though, we could do a lot more, but we would need, of course, far more stronger country commitment, particularly among the large consumers of antimicrobials, both in the healthcare delivery and in the food system. We are very chary about pointing out that, frankly, that in global dialogues. And I do understand, because most of these organizations also have member states, and they must be responsive to their member states. And so it's not a matter of, and they have donors. But I think it's up to, thanks to the efforts of those around the table, the innovation that we bring forward, the sort of public, of course, storytelling that we tell, and importantly, trying to direct that in a way that enables us actually to make it easier to persuade policymakers to take the right actions at the right time, because they're under a lot of pressure. And one should not underestimate the incentives actually to not do the right thing that we must correct.
Mm-hmm.
Well, we don't even have actually effective bans on growth promotion, which is the use of essentially antibiotics to fatten pigs and livestock yet. And this is not something that's been like unknown for a couple of decades at least, right? So there's a lot of work to be done.
Mm-hmm. Yeah. Oh, thank you very much. Yeah, and it makes us really think about all the effort we still need and areas to focus. And I have another question for Martha. How close are we to having phage products available for use? And do you think this therapy will ever be as accessible as antibiotics?
Yeah, if I can answer the second question first, I think absolutely it should be. At the moment, bacteriophages are only used right at the very, very end of those healthcare pathways where they are used. So they're generally used when most other things have failed. And phages, this is particularly in North America and in Europe, where they can be accessed. It'll be where the antibiotics have come to the end of the road. So whereas if phages could be embedded within primary healthcare, there's no reason why they shouldn't be developed as adjuncts to antibiotics. We've shown, and many other people have also shown, that you can use phages to really make antibiotics much more effective. You can use them they synergize beautifully together. So, and conceptually, they should not be expensive to produce. So you have this whole technology that's just sort of sitting there, and then the reason why it hasn't things that have limited their translation have largely been the complexity surrounding the anti-infective market that Anthony sort of alluded to. That it's a very difficult environment for people to make a quick turnaround on profit in. And bacteriophages don't sit in the normal ecosystems necessary for drug development. But I think that you can see different countries having much better joined up phage plans. So the Australians, for example, have now got a very good plan and they're rolling phages out more widely. And then in terms of products in agriculture, there's some good news. As of 2 weeks ago, there's a Polish company, funnily enough, that's just got a phage product available to use in poultry to stop salmonella in poultry. That's been approved by the European regulators. So there are products on the market. There are also products already. And the US phages have what's known as generally recognized as safe or GRAS status. So you can also buy products there. And in Southeast Asia as well, there are products that are on the market already for use in agriculture. Their use in humans is more challenging. They are used, I should say, in Georgia, which I mentioned. There are products. You can go to the Eliava Institute in Georgia and buy phages and use phages. There are some, not many places, but places like Georgia where there are products available. But it's hard to regulate those products under most regulatory systems, which has limited their pull through. So sorry, it's quite a long answer, but you can see there is traction. They should be possible. There's a lot of work to do around that regulatory piece and around the production piece as well.
Yeah, it's nice to know phage was approved for use as well, so like by the European Union. So that's nice. So another question, I think I have time for 2 more maybe. For Misbah, what has been the most powerful or surprising story you have encountered that illustrates the real world impact of antimicrobial resistance?
The story, I think obviously the neonatal sepsis story that I've mentioned already, but a story that I haven't mentioned would be the superbug threat in Gaza. I obviously couldn't visit, but managed to get through my own network a group of younger doctors that are still working in the field and reporting back. And they were sending me live pictures of osteomyelitis that had been drug resistant, either anecdotally, but also confirmed through private labs are still managing to operate somehow in Gaza. And so you had, like, it was kind of pulling a thread, and then a number of doctors started sending me pictures of drug infections that they just couldn't handle anymore. And if you're triaging, so you had obviously patients that were dying because of the explosive injuries, but then the second batch of patients, they were dying of their infections, but because it was longer waiting, then they weren't being seen in an emergency, which would have obviously not happened if it was in somewhere like Europe. But it was one of the most sort of powerful impacts, which again you knew from the Iraq War as well, when you heard about the Acinetobacter stories that you heard from. Again, that was only really taken seriously because you had US soldiers rather than Iraqi citizens that were infected by Acinetobacter. In this case, it was sort of, you know, you knew that Israeli soldiers were also being infected with something from, like, with those infections that they were picking up from Gaza, but then the Gazans themselves on mass scale. So it's kind of like, if they weren't dying due to bombing, they were dying from drug-resistant infections, and now it's starvation. So I guess that would be the most obvious story I could probably relate. And then I've heard similar circumstances in Syria of drug-resistant infections after having, you know, with the regime change now being a massive issue in hospitals over there as well. And Ukraine, like, you know, multiple, but the WHO actually did a very good job in terms of setting up microbiology labs and doing surveillance and trying to get an idea of what's happening in terms of the drug-resistant infections in Ukraine. So, you know, it's not impossible to monitor these things. MSF are doing an amazing job with the capacity that they have in terms of monitoring them as well. So in trying to get the antibiotics in, but with Gaza, the fact that there's the blockades in terms of humanitarian aid means that the antibiotics that they do need access to aren't getting in, and they're having to rely on things like vinegar to disinfect wounds. So yeah, it's a very particular set of circumstances.
Yeah, I think you published part of it already, right? I think I read it. Yeah, which is really unsettling and yeah, sad. I think, yeah, I'll ask a final question that are going to be probably direct to all speakers your thoughts on how we as scientists, bioscientists, can contribute to all sectors of the AMR and all parts of the AMR, the fight against AMR.
Yeah, I think that as a scientist, we very often sit within our science. We don't even really talk to other types of scientists. We talk to, phage biologists, but I think we definitely need to break the silos. We need to talk more to both to other types of scientists, to healthcare providers, to the general public. So I think especially junior scientists, they somehow, you don't realize how much of a role you can play by teaching, even teaching, for example, doing outreach work in schools to help support high school teachers, primary school teachers, even to teach science at an early level. Because I think if people have that interest and then the awareness, then that sort of can grow a more educated population. Because I think we need to, the public need a lot more awareness in general about science. Like there's so few of our politicians and policymakers actually have that science background. So I think scientists, by talking more to all of the different, everything from children to politicians, and I think probably I would say that younger scientists can probably make much more of a difference than they think by engaging and talking to more people in different ways.
Mm-hmm.
I could add probably that, at home, I think in their institutions, I think there's much they can do. One is, they can help translate the stories from their areas actually for the public. And they can give actually those stories vector, because the vector has both magnitude and direction. And oftentimes what we need to do is figure out how does that amplification of awareness also is matched by the right direction towards action. They also are well positioned oftentimes to re-engineer how our healthcare delivery and our food systems work. They are bringing the innovation that also can make the cost saving and other sort of changes that enable actually policymakers or give them incentive and impetus to act. And finally, they can help us also unpack the economic case oftentimes required to make their area work so they can contribute effectively to AMR. And we need scientists at the policymaking table. So those who are on the call, I hope, will actually consider that, because I know it's a sacrifice sometimes when you're trading off against what you're doing in your main day job, but we need to hear those voices at the policymaking table. You are so important to the future of this effort.
Yeah, I mean, it would make my job so much easier if all of the above happened, because half the time I'm chasing after one bioscientist that will explain one side of something, and then the others that will, and then it's like, I'm not qualified to join up the links, but I think that works, and then I'll ask someone more senior, and so it makes a very prolonged reporting process, having to, do that transdisciplinary side of things. I mean, even now to do the food and agriculture reporting, my editors are so keen on me to do more investigations into the overuse in the animal sector, but there's no data that I can show that this is directly linking to more antimicrobial resistance in humans. And when I speak to people, I'm like, we're not sure if it is. I'm like, well, it'd be great if someone did a transdisciplinary study that would show me those drivers, right? And then it's easier to pitch to editors in terms of, because they're definitely not interested unless, like, they tell me to take out big acronyms out. I mean, one of the reasons I love this beat is because I can appreciate it due to my biomedical background, but if I didn't necessarily have that, a lot of this reporting and the details and the nuance that's so important to report on this responsibly and not in an inflated way would not be there. So maybe even consider other professions, or, even if you do have a biomedical background and you want a bit of a break, try writing for other outlets and get that more, like, both Anthony and Martha have alluded to, it's like engaging more with the policy world, but also engaging other worlds as well, because it's a wealth of knowledge that then doesn't necessarily make its way out of journals. And so it would make my life a lot easier. And get in touch as well if you think there's a story that I need to cover or I need to consider. Always open to that as well.
Mm-hmm. Thank you very much, everyone, for your thoughts on these questions. And yeah, we don't have time for more questions, but the discussion was really good, really amazing. And thank you again, Misbah, Professor Clokie, Professor So, for participating in this webinar, for the amazing talks. And thanks everyone for attending. And you can continue the conversation online. Just follow at BiochemSoc on Blue Sky and X. And we welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. If you have any idea for a webinar in 2025, we invite you to submit a proposal for an upcoming webinar. You can find more information about the webinars at biochemistry.org. And all of our upcoming webinars are listed on our website as well. And if you have missed any of the 100+ webinars that we've already run as part of the series or would like to watch them again, please visit our website or YouTube channel. And the recording from today's webinar will be also available to watch within the next couple of weeks. And finally, I want to highlight that it's more important than ever to stay connected and engage with your fellow molecular bioscientists. Join the Biochemical Society community of researchers and specialists and take advantage of key benefits, including discounted registration fees for society meetings and courses. I personally recommend the practical guide for Python for beginners and R for biochemists. And you can also have exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and more. Visit their website to find out more. And thank you again, everyone, and goodbye.
Bye. Thank you.